Hi everybody, my name is Dongpei. I'm going to talk about principle of microbial microgenomics. Let me introduce that. Microbial microgenomics is the discipline of studying the genomes of microbial communities in the environment by extracting DNA directly from environmental samples and analyzing the genomes of microorganisms using high throughput sequencing techniques. Without relying on traditional cultural methods, the discipline involves steps such as sample collections, DNA extractions, sequencing, and data analysis, and aiming to reveal the compositions, function, and interactions of microbial communities, and to advance research in the fields of microbiology, ecology, and medicine. Thank you.
Now I move on to environmental monitoring and ecology. Environmental monitoring metagenomics uses sequencing to analyze all genomic material in a sample. There are two main methods for studying microbial communities. First is whole genome shotgun metagenomics. It aims to analyze all genomes in an environment to assess microbial biodiversity and function, revealing the full genetic diversity of a habitat. Second is marker gene analysis. It sequences gene-specific regions to assess the diversity and composition of taxonomic groups in environmental sample. K-marker genes include 16S ribosomal RNA for archaea and bacteria, ITS for fungi, and 16S ribosomal RNA for eukaryotes. These both methods have set new milestones in microbial ecology. Second part will be about microbial ecology. Many microbial ecology studies now integrate multi-omic data to gain a high resolution overview of community structure and function. This methodology enables systematic measurement of microbial consortia and the detailed analysis of community-driven processes, forming the foundation of ecosystems biology.
I'm going to be talking about the human microbiome and its role in health. So the human microbiome is actually the gut microbiome, and it's essential for the breakdown and digestion of certain foods that the human body cannot process by itself. It is also responsible for the synthesis of certain vitamins such as vitamin K and vitamin B. It also helps to train the immune system to differentiate between the harmful pathogens, the harmless substances, and prevent overreaction that can occur inside the body. It helps to maintain a balanced immune response by interacting with immune cells, ensuring proper immune tolerance to reduce the risk of chronic inflammation and autoimmune diseases. It is also involved in the body's metabolism by influencing how nutrients and drugs are absorbed and processed inside the body. Lastly, it also affects drug metabolism. It influences the effectiveness and side effects of medications. Now I'm going to be talking about how the gut microbiome influences responses to cancer immunotherapy. So based on a study done in Harvard, they actually conducted an experiment by where they seeded the gut microbiota from cancer patients into the mice. So based on this research, they discovered that the gut microbiota actually played a huge role in shaping the behavior of immune cells, specifically the antigen-presenting cells, that is important and crucial for detecting and fighting cancer. So what they gathered is the mice with the microbiota from successful patients actually had lower levels of PDL2. Now that is a molecule that inhibits immune activity. However, the mice with microbiota from non-responding patients had higher levels of PDL2. Now that actually dampened their immune defenses against cancer. An important discovery from this research that a specific bacteria called Bacterium C. cateniformis actually helps to lower PDL2 levels. So this actually helps to make immunotherapy more effective. So this suggests that the gut microbiomes could actually be used to improve immune responses against cancer. Now this is a possible new approach to a new treatment option for cancer immunotherapy.
Let's talk about these applications in agriculture and food productions. Microbial metagenomics has revolutionized agriculture and food production by harnessing the power of microbial communities. In agriculture, it improves soil health by identifying beneficial microorganisms to improve nutrient cycling, paving the way for less reliance on chemical fertilizers and sustainable agriculture. It also promotes plant growth by discovering plant growth promoting bacteria that can be used as biofertilizers to increase crop yields and stress resistance. In addition, metagenomics supports disease management through early detection of soil-borne pathogens and implementation of biocontrol strategies. In food production, Metagenomics optimize the fermentation process of products such as cheese, yogurt, and wine, ensuring high quality and consistency. It enhances food safety and reduces foodborne risks by identifying spoilage organisms and pathogens. In addition, it unlocks novel enzymes and bioactive compounds that facilitate food preservation and processing. Together, this application demonstrates metagenomics precision as a cornerstone of innovation in the agriculture and food industries.
So for the application of microbial metagenomics, there are two parts. The first one is the biotechnology part and the second is the industrial part. For the biotechnology part, let's first look into antibiotics. Metagenomics has advanced antibiotic research. Soil microbiome studies have led to the discovery of antibiotics like streptomycin. Functional metagenomics identifies new antibiotics and resistant genes, while descriptive metagenomics tracks resistance in the environment, helping combat global health challenges. Moving on to enzymes, metagenomics has unveiled enzymes like lipases and proteases, essential for industries. These enzymes power high-performance detergents, degrade polymers like cellulose, and synthesize complex chemicals, driving sustainability and innovation. Moving on to the industrial applications, you can first see agriculture. In agriculture, metagenomics helps us unlock the hidden potential of soil microbiomes and also enhances soil fertility, fixes nitrogen, and promotes crop growth. It also helps in developing natural biopesticides, all in all paving the way for precision agriculture where farmers can cultivate healthier crops while reducing chemical inputs. Moving on to the food industry, metagenomics immensely benefits the food industry. It aids in identifying microbes for improved fermentation process, ensuring better quality and taste in products like yogurt, cheese, and beer. Moving on to the environmental monitoring. Metagenomics is vital for environmental monitoring and sustainability. It helps us track changes in microbial communities, assess pollution levels, and restore ecosystems. For instance, metagenomics identify microbes that break down plastics, degrade oil spills, and detoxify heavy metals, offering eco-friendly solutions to some of our biggest environmental challenges. Now, if you've noticed, I've skipped 2 points in each slide. Biotech compounds and pharmaceuticals, because this is where the industrial and biotechnology applications of microbial metagenomics connect. In pharmaceuticals, microbial metagenomics is a game changer. Scientists can uncover novel genes for antibiotics, antivirals, antifungals, combating the global threat of antimicrobial resistance. Metagenomics also enables the discovery of bioactive compounds like terragens, biolacin, and injurimin. These discoveries have led to the life-saving drugs such as cytarabine for cancer, cephalosporins for infections, and vitarabine for viruses, proving the immense pharmaceutical potential from marine microorganisms.
Now I'll be talking about the evolutionary and functional studies of microbial metagenomics. First, it's the evolutionary studies. Evolutionary genomics refers to the study of how features or components of a genome change both within and between species over evolutionary timescales. The interpretation of any changes in the structure or content of genomes is often made within a comparative phylogenetic context in order to account for the underlying noise in the data as a result of the variability of DNA sequences due to a population genetic forces. Some common questions in evolutionary genomics are: how and why does the total size of genomes change? Why do some species have larger genomes than others? How do genomes or parts of genomes change in size during evolution? Why do some species have more coding DNA than others? And so on. Now I'll be talking about the evolutionary and functional studies of microbial metagenomics. First, it's the evolutionary studies. Evolutionary genomics refers to the study of how features or components of a genome change both within and between species over evolutionary timescales. The interpretation of any changes in the structure or content of genomes is often made within a comparative phylogenetic context in order to account for the underlying noise in the data as a result of the variability of DNA sequences due to a population genetic forces. Some common questions in evolutionary genomics are: how and why does the total size of genomes change? Why do some species have larger genomes than others? How do genomes or parts of genomes change in size during evolution? Why do some species have more coding DNA than others? And so on. Now let's move on to the functional studies part. Functional metagenomics involves isolating DNA from microbial communities to study the functions of encoded proteins. It involves cloning DNA fragments, expressing genes in a surrogate host, and screening for enzymatic activities. Using the function-based approach allows for the discovery of novel enzymes whose functions would not be predicted based on DNA sequence alone. Information from function-based analyses can then be used to annotate genomes and metagenomes derived solely from sequence-based analyses. Thus, functional metagenomics complements sequence-based metagenomics analogs to how molecular genetics of model organisms has provided knowledge of gene functions that is widely applicable in genomics. Thank you.
Okay, that concludes the end of our presentation. Thank you for watching.